Oh, I just realized now that I'm in presenter mode, I don't have a clock. So I'm just gonna look over there once in a while. All right. Hello, everyone, and welcome to the Society for the Study of Reproduction Rising Star webinar. My name is Andrew Modzelewski. I'm an assistant professor at the School of Veterinary Medicine here at UPenn, and I'll be your moderator for this webinar. We have two rising stars who will be presenting their work for you today. But first, there's just a couple of housekeeping items, SSR business before we get started. All participants are muted with the exception of the speakers and myself. The webinar is scheduled for 60 minutes and it's being recorded. It will be posted on the SSR website pretty shortly after today's webinar for members to access at their convenience. Please feel free to submit your questions during the presentation using the Q&A button on the bottom, not the chat button, the Q&A button. The chat button is for technical questions if you can't hear the speakers or hear me. And that's at the bottom of your screen. And during the question and answer section, I'm going to read them out. Specifically, if there's any clarification, I'll call that person up. If you have any questions or concerns about this webinar, feel free to reach out to us at the SSR headquarters via info@ssr.org. And with that, let's get started. And we're going to meet our first speaker. Devanshi Jain is an assistant professor coming to us from the Department of Genetics at Rutgers. Devanshi did her postdoc in the lab of Scott Keeney at Sloan Kettering, working on some of our favorite genes like the DNMTs and characterizing new ones like KET2 and RAHU. The Jain Lab is interested in how dynamic gene expression programs that drive gametogenesis are controlled and what the roles of post-transcriptional RNA regulation are in the orchestration of meiotic events. And something that I'm particularly interested in is their work on genome defense mechanisms that repress retrotransposons in the germline and their coordination with other chromosomal processes such as meiotic recombination. And with that, let's hear from Devanshi about TEX14 and intercellular bridges and how they bridge the gap between germ cell connectivity and male fertility. But real quick, Devanshi, can you unmute yourself and just make sure we can all hear you? And if you're still quiet, we're going to get to technical services to fix that for us.
All good?
All right, I hear you. If anybody can't, please use the chat or forever hold your peace. All right, Devanshi, take it away.
You'll have to stop share, Andrew.
Yeah. You got it.
Okay. All good? Shall I start? All right. So my lab is interested in the cell differentiation process that generates gametes. And most of our work is really centered around understanding meiosis. So this is the specialized cell cycle that halves the genome complement so that when a sperm fertilizes an egg, diploidy is restored. And here I'm just showing you meiosis cartoon on the top with mitosis at the bottom for comparison. And a pair of homologous chromosomes is shown in blue. Now, in both meiosis and mitosis, you have one round of DNA replication. In mitosis, this is followed by one round of chromosome segregation. However, in meiosis, this one round of replication is followed by 2 rounds of chromosome segregation, and that is how diploidy is halved. Now, critical to segregating your chromosomes properly is an event called meiotic recombination. So this is the deliberate formation of hundreds of genome-wide double-strand breaks that are then repaired by homologous recombination. So homologous recombination between mom and dad chromosomes, of course, creates genetic diversity, but more importantly, homologous recombination between mom and dad creates these physical linkages between the chromosomes, between the homologs, that helps orient them on the spindle and is absolutely essential for chromosome segregation to occur properly. Now, one other thing I want to tell you for this talk is the formation of the synaptonemal complex. This is a proteinaceous structure that forms between the homologs and holds them together and is essential for meiotic recombination to proceed properly. So in my lab, we're really interested in meiotic recombination pathways and how they maintain germline genome stability. And like Andrew said, we're also really interested in meiotic gene expression. So we want to know how meiotic RNAs that encode for proteins that will do all the critical meiotic events, how are these RNAs synthesized, stabilized, degraded, and translated? So today's story actually starts with a random mutagenesis screen in mice that I employed as a strategy to explore mammalian meiosis while I was still a postdoc with Scott Kinney at Memorial Sloan Kettering in New York. And so this is a very brief overview of my screen. I randomly mutagenized mice using this point mutagen, ENU, and the hope was that I would potentially isolate interesting alleles of genes with point mutagenesis. I then bred mice for 3 generations to homozygose the mutations I'd induced. Then I screened third-generation mice using a variety of different readouts. And for mouse lines that had an interesting meiotic phenotype, I identified the potential causative mutation using SNP discovery pipelines that were tailored to my screen. So this essentially allowed me to gather a collection of point-mutated alleles of genes, many of which were either completely unexplored or very poorly understood in the context of meiosis. And they've served as starting points for projects in my lab. And today's story starts with one such mutant that we call the TAMA mutant that showed a defect during meiotic prophase, specifically in meiotic recombination, so the formation and repair of breaks. Now, I was cytologically screening for meiotic defects using these 2 markers. So CYP3, the first marker, is a synaptonemal complex component. So remember, a synaptonemal complex is the structure that holds the homologous chromosomes together. And what's important here is that CYP3 undergoes visibly distinguishable changes in staining patterns as cells progress through meiotic prophase. So you can use it to stage cells. And then the other marker I was using was gamma H2AX. This is phosphorylated histone H2AX. It's a break marker. So I was using it to monitor break formation and repair. So in early prophase cells, when breaks are forming, it shows pan-nuclear staining. And in late prophase cells, when breaks are predominantly repaired, it's gone from most of the nucleus that contains the autosomes, and it's restricted to this small region that covers the sex chromosomes. And this is what these stainings look like in nuclei squashes from wild-type testes. Now, if we count the proportion of cells that are in early prophase-like stages forming breaks, and those in late prophase-like stages that have repaired breaks, this is what the distribution looks like in wild type. Now in TAMA mutants, what we noticed was that late prophase cells were depleted. Additionally, there was an accumulation of abnormal cells where CYP3 staining patterns suggested that these cells were trying to enter late prophase, but gamma H2AX was still retained. So this is a classic break repair defect phenotype. And the point mutation mapped to this gene called TEX14. And we know we mapped the mutation correctly because the TEX14 null allele fails to complement the point mutant. Okay, so what is TEX14? So Marty Marzuk's lab showed several years ago that TEX14 is an essential component of germ cell intercellular bridges. So TEX14 is expressed in germ cells where it localizes to the mid-body of dividing germ cells and prevents important protein-protein interactions from happening, thereby preventing abscission. So what it ends up doing is it ends up causing formation of an intercellular bridge between germ cells, and it's retained at this intercellular bridge as a structural component. And here you can see TEX14 staining in between two adjacent germ cells. Okay, now before meiosis, germ cells undergo mitotic amplification, and intercellular bridges form during these mitotic amplification cycles and during the two meiotic divisions. And these bridges persist until the very last stages of spermiogenesis. So they're pretty stable. They're also pretty wide, and RNA and protein have been shown to move through these bridges. So from a meiosis-centric viewpoint, what this really means is that meiosis actually occurs in a syncytium. So this is true in mice, but not just in mice. This is a conserved feature. So meiosis occurs in a syncytium in flies, even in worms. What is the role of this is not fully understood. And the best understood example is from female flies, where germ cell connectivity, so the syncytium, plays a role in nursing. So what happens in this model organism is that many germ cells donate their cytoplasm to one germ cell that becomes the future oocyte. This is the nursing mechanism. But male mice do not utilize a nursing mechanism. However, Marty's lab showed that these TEX14-mediated intercellular bridges are actually required for spermatogenesis, so for fertility in males. And this is what they actually showed. So in the absence of TEX14, germ cells lack intercellular bridges, so they're individualized. Now, in females, this individualization does not drastically impinge on fertility. However, Diana Laird's lab showed that this does result in uncoordinated meiotic entry for these germ cells, but the cells do meiosis. Marty's lab showed that in males, however, these individualized germ cells have problems later on during meiosis, and as a result, they are infertile. Okay, so coming back to the point mutant. The point mutation that we isolated lies in a splice site region encoding the C-terminal portion of TEX14, and it translates as a drastic reduction in protein level compared to wild type. However, compared to the null, some TEX14 protein is present. And we think that this protein forms reduced intercellular bridge-like structures. So we can look at these intercellular bridges by staining for TEX14. And what you're seeing here are hole-mounted seminiferous tubules stained for DAPI and for TEX14. And so in wild type, what you see are these TEX14 positive structures between adjacent DAPI positive cells. Now in these TAMA mutants, in the point mutant, we can find instances of TEX14 staining between adjacent cells. We can also look at bridges through EM. So what you see in wild type is this electron-dense channel that connects to adjacent germ cells. And in point mutants, we can see such channels connecting some germ cells. So you see an example there, and here an example here. So we think it's like a hypomorph. And remember, I told you that these point mutants fail to complete meiotic recombination. As a result of meiotic recombination defects, the meiotic cells die, and these mice are infertile. But if we compare the null, the TEX14 null, to the point mutant and look at the presence of meiotic cells within the seminiferous tubules, what you'll see is that in this TEX14 point mutant, there are a lot more meiotic cells present compared to the null. So this fits with it being a hypomorph. So we essentially decided to use this hypomorph to ask this question: which meiotic mechanisms depend on germ cell connectivity and intercellular bridges? So I'm just going to summarize here that the original paper did not identify any gross differences in the TEX14 mutants in the spermatogonia, so in the mitotically dividing germ cells prior to meiosis, and neither did we. So we focused our analysis on meiosis. And we first asked, are these individualized germ cells, the bridge-deficient germ cells, actually able to do meiotic replication, the very first thing the cells need to do when they enter meiosis? And we looked at replication by injecting mice with the nucleotide analog EDU, which we can stain for, and we co-stained for a marker that's present when cells are doing meiotic replication. So that's striate. And what we see with these markers in wild type is that adjacent striate positive cells evenly incorporate EDU, okay, shown in green. However, in the point mutants, what we saw was that there were many instances where you had striate positive cells, that's in pink, that had not incorporated EDU. So what this was telling us was that these cells were having trouble doing meiotic replication. Now we can also look at replication in bulk by FACS sorting for meiotic replicating cells using these markers, and then basically inferring replication status through DNA content. So when we do this, what we see in wild type is that most cells are either replicating or have just finished replication and entered prophase. However, in the point mutants or in the null, what we found was that many cells were stuck in a pre-replicative state. Okay? So our interpretation of this was that in these TEX14 mutants, when germ cells are individualized, cells have a problem in undergoing meiotic replication. If you are not able to do meiotic replication, you are not going to enter meiotic prophase, and of course, this leads to infertility. However, some cells were entering prophase. So we next asked, how are the prophase events proceeding? Now, remember I told you we actually isolated these point mutants as mutants that could not do meiotic recombination properly. So we looked into this in more detail, and I'm going to show you just two pieces of data. And the first one is looking at CYP3 and gamma-H2AX staining. This is the same staining I showed you before. So CYP3 is a synaptonemal complex component, and gamma-H2AX is the break marker. But this time, we're looking at chromosome spreads. So the chromosomes are splayed out on a slide. And we're looking at late meiotic prophase. So at this stage, break repair is expected to be near complete. So what we see on wild type is that this break marker gamma-H2AX is not present on autosomes and is only restricted to this small region that covers the sex chromosomes. However, in the point mutants, there were several instances where chromosomes had fully synapsed, which we can tell from the CYP3 staining. However, gamma-H2AX was still retained. And we know that these chromosomes also retained recombinase foci, so foci of RAD51 and DMC1. So we know that these cells were not being able to fully repair their breaks. Now, the other piece of data I'm going to show you is staining for MLH1. This is a marker for crossovers. And remember I told you crossover, so the physical connections between homologous chromosomes is absolutely essential for chromosome segregation to proceed normally. And so every chromosome pair needs to have at least one crossover. And what we see in wild type is that all chromosome pairs have these MLH1 foci, so they have crossovers. However, in the point mutants, there were several instances where chromosomes pairs lacked these crossovers, okay? So there was reduced meiotic crossovers, and of course, as expected, this would lead to problems during meiotic metaphase, and this would lead to chromosome segregation errors or cell death, and therefore infertility. So what this data was telling us was that these TEX14-mediated intercellular bridges are important for meiotic recombination repair, right? So for meiotic recombination to proceed properly. Now there is one other threat to genome stability that these cells need to contend with, and that is transposons. So transposons are abundant in the genome, and germ cells have adopted many repressive mechanisms that keep them in check. However, meiosis is especially vulnerable to transposon derepression, and that is at least in part because one of these transposon repressive mechanisms is stripped away during meiosis. And so this is partly the reason why mutants of transposon repressive mechanisms manifest a phenotype during meiotic prophase. So we asked whether transposons were being repressed properly in these TEX14 mutants, and we did that by staining for ORF1p, which is a line 1 retrotransposon encoded protein. And strikingly, what we saw was that in these point mutants, meiotic cells, which are stained in green, accumulated high levels of this retrotransposon encoded protein. Now we could detect retrotransposon RNA through FISH and even through RNA-seq, and I'm just going to show you single-cell RNA-seq data. So here what you're looking at is the trajectory in wild type and in the point mutants. And in the point mutants, all the late germ cell stages are absent, just as expected. But what's important is that if we look at differential gene expression between wild type and the point mutant, we don't really see many drastic changes during early germ cell stages or during early meiosis. But in late meiotic stages, we see a massive upregulation of transposons and gene deregulation. Now, since we had single-cell RNA-seq data, we actually used it to ask this question. In these disconnected germ cells, in these TEX14 mutants, is cytoplasmic sharing actually disrupted? And we looked at that by looking at haploid spermatids. So haploid spermatids from wild type contain transcripts from both X and from Y, even though they're haploid. So they only contain either the X or the Y chromosome. So this has been seen before, and this was as expected. This suggests that there's sharing of cytoplasm going on between the haploid spermatids. Now, our expectation was that if sharing is truly disrupted in these TEX14 mutants, now the few cells that reach the haploid spermatid stage should contain only X or only Y transcripts. And that is actually what we saw. So you see there was abundance of haploid spermatids that had only X or only Y. But of course, remember, this is a hypomorph. So there were some instances where cells were still connected and contained X and Y transcripts. Okay, so just to kind of wrap up what I've been telling you, what we think is that this individualization of germ cells disrupts meiotic events in many ways. Firstly, these cells are not able to undergo meiotic DNA replication, and that leads to cell death and infertility. But cells that do manage to replicate and enter prophase are not able to repair meiotic breaks, and they massively de-repress retrotransposons. So our interpretation of this is that germ cell connectivity is playing a role to maintain genome stability. And we'd love to know the exact mechanism through which this is happening. One obvious hypothesis would be that this is happening through exchange of some regulatory factors. And we do not know at this point what those regulatory factors are, but something that we would very much be interested in finding out in the future. We're also really interested in finding out how germ cell connectivity actually influences meiotic replication specifically. So we know meiotic replication compared to mitotic replication is special. We know it's very slow, we know there are many differences, but we don't know all the different ways in which it is special. And it looks like germ cell connectivity may be important for specifically meiotic replication to proceed properly, and we'd like to know exactly why. Okay, so with that, I just want to highlight the people that did the work that I presented. So Julia, Kevin, and Shreya in my lab did the bulk of the work that I talked about. And it would not have been possible without some amazing collaborators. So Flor Prato at the NIH helped us with replication analysis. And Mashiat Rabbani in Sue Hamoud's lab, and Matt Layler in Chris Ellison's lab at University of Michigan and at Rutgers were critical to doing the single-cell RNA-seq. And thank you for your attention. And with that, I'll take any questions you may have.
Okay, Devanshi, thank you very much. Very, very interesting stuff. Thank you for presenting it so clearly. Okay, so we're starting to get some questions here. Okay, I will moderate and I will ask the first question. So we have an anonymous attendee who's asking, good talk. Why are some sperm with point mutations still entering the next stage, but some just don't? So what is maybe the critical factor that's preventing this progression?
So my understanding of the question is that why in the point mutant, we have some cells progressing to the next stage. So I just want to clarify that the point mutant is a hypomorph and not a complete null. And by that, what I mean by hypomorph is that some amount of connectivity is still present, whereas many of the germ cells are disconnected. So that would be the reason why in a hypomorph, you would have cells progressing to later stages compared to like a gene null. I hope that answers it.
If there's a follow-up, you could type it in the chat, in the Q&A. All right, next question from Grant McGregor. we hear, couldn't it also be possible that text 14 functions independently in recombination versus control of abscission?
You know what? We actually cannot exclude that possibility. So it is in theory possible that text 14 is doing something that's independent of its main function in forming bridges. But I'll just sort of add here that, we don't see, we and others who've tried don't see by immunofluorescence, TEX14 going anywhere else in germ cells. So that's why I think this possibility is unlikely. It would actually be really hard for us to disentangle this because, potentially require some sort of a mutant situation where TEX14 is dysfunctional, but bridges are still intact. And then we'd be able to ask, okay, TEX14 is dysfunctional, but bridges are still intact. Is meiotic recombination occurring normally or not? But I totally agree with the person who asked the question that I mean it could be, and we cannot exclude that.
All right, we got one from Kyle Orwig. Hey Kyle. I guess half of the germ cells could contain X and TEX14. Are you getting a half level of sperm production, or are the bridges equally important?
I don't fully can you repeat that, Andrew?
I'll repeat it. I guess half of germ cells would contain X and TEX14. Are you
So
getting a
does that
half?
mean X transcript, like X chromosome?
Kyle, would you like to follow up? But so I'll just finish the question again. Are you getting a half level of sperm production, or are their bridges equally important? So yes, X chromosome.
So in the question would be like in the hypomorph, are we getting half the number of like 50% reduction in
Production, yeah, sperm production.
in sperm production? It's a lot. I mean, if we look at the proportion of spermatids, so the number of germ cells that make it all the way to the spermatid stage, it's more than a 50% reduction. So I don't know. It's hard to know how many cells need to be connected to actually reach the spermatid stage. Let's say, for example, 100 cells need to be connected, right? So we don't know in the TEX14 mutant, did that 100 get reduced to 50, or is it just will every cyst be affected the same, or is it sort of little like kind of random, right? So whether that one cyst successfully makes it to a certain number of spermatids, I mean, I don't know how I would actually even like get to that.
Yeah, and then Kyle followed up with hypermorphs complicate interpretations, which is
does, yeah.
for sure. Thanks, Kyle. Cindy is asking, do you find that TLR7/8 differentially expressed in the X and Y spermatids in the single cell RNA-seq? The question is related to recent reports of using them for sperm sorting.
There's many different, for the cells that do reach the spermatid stage, there's many differentially regulated genes. I don't remember off the top of my head if that is on the list, but I mean, I could check later and get back.
I mean, by the way, everybody, if you have follow-up questions, we encourage you to reach out offline or through your own email to contact the speakers. All right, are there any more questions? I guess I sort of, oh, here we go. Okay, here we go. Grant McGregor, do other mutants that affect intercellular bridges also affect recombination?
That's a really good question. So I don't know of another mutant that will remove intercellular bridges during early during meiotic replication or during when breaks are forming, or I mean, if somebody else knows of one, I'd love to hear. But yes, that would be another way to kind of get at what somebody else was asking like earlier, that if we had another mutant that could specifically only remove bridges, we would be able to see what the effect of that is.
And then finally, Anonymous asks, does hypermorph mean the variation?
Does hypomorph mean the variation?
Yeah.
I'm sorry, I don't understand the question.
That's all we got. If Anonymous wants to clarify, feel free. If not, you can always ask Devanshi directly. But we are running out of time here, so I do want to make plenty of time for Miguel. And Devanshi, thank you so much. That was excellent, very interesting. I knew about bridges in meiosis, female meiosis. I wasn't fully aware of them in male meiosis. It's very interesting. I wonder if there's when you break the bridges, there's this sort of X-ness and Y-ness to them. But this is very, very fascinating stuff. All right, cool. So we're gonna switch over. I'm gonna jump back in. Let's kick you off the share screen. I'm gonna kick myself back on. Yes. All right, I'm hoping you guys could see the correct screen. If not, we'll get over it. But all right, so up next we have Miguel Angel Brieno Enriquez. And I'm sure I messed that up, but I'm also sure that Miguel is not going to correct me. Miguel is also an assistant professor in the Department of Obstetrics, Gynecology, and Reproductive Sciences at the University of Pittsburgh at the Maggie Women's Research Institute. Miguel did his postdoc with my PhD advisor, Paula Cohen, and also worked with Ned Pleiss, co-mentored at Cornell University, looking at various molecular aspects of meiosis, including the cohesins, NEC1, telomeric repeats during meiosis gonad development. Miguel's lab shifted to looking at the model, fully shifted to looking at the model system of an extremely long-lived species, the naked mole rat, in order to better study reproductive aging in mammals and human meiosis in general. And now let's hear from Miguel about what we can learn from one of the cutest species, the naked mole rat, about AIDS-associated decline in fertility. And with that, Miguel, if you could take it away.
Okay, give me a second.
Oh, take your time. We have 30 seconds.
Sorry. Okay,
All right. It
can
looks
you hear
good to
me?
me. Take it away.
Okay, perfect. Thank you, Andrew, and thank you everybody in the committee for this invitation. It's a real honor to be here to share our research with SSR community. So during the last 150 years, the lifespan in human has been almost duplicated from going around 40 to 150 to 80 years, let's say. However, during this 150 years, when it comes to reproductive lifespan in females, there is this kind of plateau where in the best case scenario, menopause will come and knock your door at 51 or 52. There's an important part of ovarian aging that in many years was only focused on the idea of the ovary, it's aging, you don't have babies. However, ovarian aging goes way beyond that, and it will be affecting every single layer of the body. Someone could say that is something really vain as a little wrinkles on your forehead, but it goes deep and deep, affecting muscle, blood vessels, bone, increase the risk of cancer, and also decrease cognitive function. And the big question here is about how this happens. And it's because, well, as you know in this room, that there's like a dogma for reproductive biology in females. And basically it says that the number of germ cells in the ovary at birth, it's all that you want to have for the rest of your life. And this is based on how is the development of the ovary or the germ cells. And here we have a cartoon representing the human ovary. Here we have the wicks, and we have initially the primordial cells that are the cells that will give origin to all the germ cells. These cells will go to a massive mitotic division to reach the maximum or number of 7 million oocytes. After that, they will go to different differentiations, getting into meiosis, getting folliculogenesis. At the same time, they have checkpoints, and this number will start like dropping. But finally, in the best case scenario, have around 1 million at birth. This doesn't stop there because it keeps going down. And by the time of the first period, only 400 of these will be there. and they will be using during these years to try to ovulate them. And finally, at 51 or 52 years, and again, if everything goes well, you're gonna have around 1,000, and menopause will be there. However, to every single rule, there's an exception. And let me introduce you the beautiful naked morat. Yes, probably you say this animal is quite ugly, but trust me, these animals already won in life. The first thing that we need to know is where they are from. And they're originally from Africa, from the region of Kenya, Ethiopia, and Somalia. And they're part of the African mole rats. These animals are really famous in the specific, the naked morat, but also one of their cousins. And they're famous because they're like one of the few mammals that are eusocial. What this means that like bees or ants, there is a queen that is in charge of reproduction, and they're like other animals in the colony that will be taking charge of other tasks called subordinates. However, what it makes different is that in the naked morat world, every girl can be a queen because they're not primed to be it. So if you remove the colony after 6 months of age, they can be a queen and they can be pregnant. Naked morat is really famous in aging because so far we know that this animal is the longest lived rodent. So far, 41 years in the lab. They have highly resistance to pain. They have modification in certain kind of terminals. They also have high survival to anoxia, 80 minutes without oxygen, and they can be happy, almost zero chances to cancer. But most important to us, they have no decline in fertility during the entire 4 years. So that means that actually they can die having babies. So we published recently that in this case of the naked morat, here we can see ovaries, postnatal ovaries going for day 1 up to 90, labeled with the marker of germ cells, VASA and DAPI, that basically the ovary of the naked morat is packed of tons and tons of germ cells. We count these germ cells and we found that there's a peak about 1.5 million of germ cells at P8. That is kind of equivalent to what we saw in human, telling us that these animals, they have a really large ovarian reserve. But also they have other peculiarities, and it's that when we were analyzing this, we were able to detect cells possibly for markers of pluripotency such as SOX2, OC4, and the marker of PGC is BLIM1. These cells are positive from P9 to P90. They tend to drop, but they never disappear. The other important part is that I mentioned that in human and mouse, there is this massive apoptosis and then drop the number from 7 million to 1. In the case of the naked morat, this happens, but in a little tiny period of time, about P15 with this peak. But even in this case, there are around 1 million cells. So saying that there's a reduced apoptosis. But more important, we found that these cells, positive for markers of germ cells, are able to divide in vivo, but also we show that it's possible in vitro. And one of the important things about this is that says that a postnatal oogenesis, but it's not just happening in young animals. When we analyze new queens labeled here as exubs and we stain it with markers of cell division as Ki67 and phosphohistone H3, we saw that there is increase in the adults, saying that there's postnatal oogenesis in adults. But what are the mechanisms? Well, for this, we need to think about all the mechanisms that the naked morat has, and they can go from genomic stability to cycle regulation and so on. But we are particularly interested in the extracellular matrix, and it's because this animal has a really peculiar one. But first, we need to know or remember that the naked morat the extracellular matrix used to be like this kind of glue between the cells that works for nothing. However, now we know that it's critical for transduction of mechanical signals, sequester and store growth factors, and obviously it's important for the structure. So, and if there's one point that is really important for us, and it's like they provide plasticity to the cells that allows that these cells move from stage 1 to stage 2 and differentiate, also to migrate, but one of the most important to give this kind of transition state where there is like a temporary and reversible movement of cells, similar to what we see in the stem cells asymmetrical division. But what is the extracellular matrix made of? Well, there are like many components like collagens, like integrins, fibronectins, and we are particularly interested in proteoglycan complexes. These are made of carbohydrates, protein, and polysaccharide. In specific, from all the proteoglycan complex, we are interested in one called hyaluronic acid or hyaluronan that is based off those ones that I mentioned, and it's in a different number. So it means that this is a block, and depending on how many blocks you have together, it will be the properties. And again, our girl, the naked morat, had like a really peculiar one that gives something called very high molecular hyaluronan that is like 6 to 12 million Daltons. And in fact, this is important because the size of the HA or the hyaluronan will be important for the function. Oligo-HA, when it's forming or when it's a product of degrading, one of these ones, has like it's really small, but also it's related to aging, inflammation, apoptosis, cancer. Instead, when we are here, that is where human and mouse normally should be, they're like tissue homeostasis and metastasis. In the case of the naked morat, it's like related to cytoprotection, anti-metastasis longevity, and we propose that it's also related to pluripotency in the case or the ovary. But what makes that the naked morat can have this massive HA? Well, HA is produced or synthesized in the naked morat ovary by hyaluron synthase 2. This is highly conserved across the species from naked morat, mouse, human, etc. And there are like two just changes in amino acids. There's this serine and this serine that confer the characteristic to create this really, really big hyaluronan. Years ago, Gorbanova lab published this and also analyzed that the expression of this HA increased the viscosity in the cell cultures. And in fact, when they analyzed by pulse field, they can see that there's like these big blobs of really very high molecular hyaluron. They also test which one of the Has are like important for these synthesis, like Has 1, 2, and 3. And they show that in this case, Has 2 is the most important one for them. So now that we know that is that thing and the HA and in the naked morat, the question is how it looks in the ovary. And for that, first we did the staining with HA binding protein, hyaluron binding protein here in green, and we can analyze them from day 56 up to 6 years old queen. We can see this all over the ovary. Later on, have like this kind of biphasic where it's in the cortex and in the medulla, and it keep going around. At 6 months, we can see in the follicles, but if you see, and same as 6 years old, but in all of them, there's this high concentration or levels of HA in the cortex. And this is important because the cells that were positive for pluripotency and PGC markers are located in that place. However, this is HA, but it's not telling us anything about who is synthesizing it or what is the size. So to ask that question, we extract HA from the naked morat ovary at embryonic day 56, 6 years old queen, and we measure it. Here have as a control positive HA from the skin, and we saw that actually it's in the right size. We're showing that we have very high molecular hyaluron. We evaluate which one of the enzyme is in charge of the production of this, and we saw that even when Has2 and Has3 is expressed, in the naked morat ovary, seems that Has2 is the predominant one, at least in these ages. Interesting, in mouse and humans, seems like Has3 in adults is the one that is controlling this. However, if I'm saying that they're like in the cortex, that they should be the cells, so we the PGC, so we decide to analyze this with the double marker of SOX2, pluripotency factor, HA, and we have this for OCT4 and BLIM1 that we can show in another moment, and we can see that like surrounded by the HA. And in the case of, for example, postnatal day 8, we see this lovely cluster that seems like a germ cell nest and is surrounded by HA. It doesn't stop here because we can see this in 6 years old queen. There's like SOX2 positive surrounded by HA, saying that probably it's important for this. But how important is this? It's just something that is related to naked morat, or we can see it in another species. So for that, first we went to our best friend, the mouse, that is one of the used ones, and we analyzed the expression of the enzymes in charge of the synthesis of HA. As we can see here, in mouse at embryonic day 11.5 to 13.5, seems like Has2 is the one that is controlling the synthesis. This is really interesting because publication from Duncan Lab show that in adult, in human and mouse, Has3 is the most important, seeing that in the fetal development is different. We detect the HA, and also we were able to see that with differentiation, because entry into meiosis is happening here, meaning that those cells are differentiating, the amount of HA is dropping, suggesting that probably is involved in entry into meiosis or differentiation of the PECs. But most important, we were able to detect this in human ovaries. So we have 9 weeks, 12 weeks, 70 weeks, and 20 weeks. And we can see that, in fact, this ovary looks more like the naked morat, where we have all over the place, but it tends to be in the cortex. We analyzed in these ovaries that, and we saw that during the development of the ovary in fetal ovaries, we can see that Has2 is also highly expressed, and then it starts to drop. To try to see where it matched with the development, we can remember this little cartoon, and we can see that there's like this peak of germ cells. So at early stages, we see that cells are SOX17 positive. This will be equivalent to SOX2 in PECs in human that is surrounded by HA, and they're like breaking apart and differentiating, and the levels of SOX2 positive as well as the HA is dropping down. Now that we know that it's also conserved across the species, is like, what's going to happen if we remove HA from there? For that purpose, we don't have in naked morat yet a mutant, work in progress. However, we can develop systems where we can get ovaries and culture them, and then analyze them for sections, single nuclear RNA-seq, meiosis. And in this case, we use this compound called 4-MU that it will be inhibiting the production of HA and also dropping the transcription of HA. So it's what we did. And the first thing that we analyzed is like, what's going to happen if we culture a naked morat ovary during 5 days with or without this component. And as you can see here in green, they have this lovely HA, is close to the cortex where like all the germ cell, like the positive for SOX2, etc. And then with the treatment, they drop down. We also see that in the pulse field, there is a drop in the amounts of HA in the group control compared to the treatment. So we decide to see what is happening here and see the SOX2 cells. And you can see again the clusters with HA, a lot of SOX2 positive. When we treat it and we drop the levels of HA, what we saw is that these cells that are pluripotent, that are PECs, are gone or highly reduced. We evaluate this, and you can see how the numbers heavily drop, indicating that these cells are either dying or differentiating. So thinking about what we see in another species, we thought, well, probably a good option is that they are differentiating. So for that, what we did is in these cultures, we analyzed the level of PECs with BLIM1, and we saw that in cells that are cultured with the treatment with 4-MU, there is a drop in the number of cells positive for BLIM1, but there's an increase of STRA8 that is a marker of entry into meiosis. So it goes from almost nothing in the control and a lot of them in the treated with 4-MU. And this is also true for the REC8, that is a meiotic specific cohesion that we can see that increase, saying that what are we seeing are cells that with the drop of HA, they're like committed into the entry into meiosis. But this is just like from the point of view of staining. So what is happening at the level of transcription? For that, we got this cells from single nuclear RNA-seq from these ovaries in culture. So we have the plot together and then separated them. And the first thing that we observed is that there are like 2 clusters that were like heavily modified. This first one labeled in 6, it's positive and disappear in the treated, is positive for the markers of pluripotency and PECs, suggesting that those cells are either differentiating or dying. But the other thing is that this orange in 2 that is positive for markers of meiosis is going to low number of cells to a lot of cells, suggesting that the results that we saw with the stains are correct, and with the absence of HA or the drop in NHA levels, we induce differentiation. And that actually match with things, reports in the stem cells biology, where when you modify the HA in the matrix, they can be differentiated. But the next question, like what happens if you have too much? And for that, what we did is to have this hypothesis that if we take the naked rat gene and put it in mouse, and we express this, that will be the equivalent of our expressor of HA, we're going to have like increasing mitotic proliferation and a large ovarian reserve and this kind of extracellular anti-aging process. So as a proof of principle, first, we create this animal with the Dr. Vera Gorbanova, where we have the Has2 flanked by a LOXP site, and then activated by tamoxifen, saying that this is going to be expressed in the entire body. With this, we took animals or 16 months, we treated with tamoxifen, we wait a month, we sack them, and we obtain the ovaries. And we saw that the amount of HA in both is really different. But more important, we are seeing the deposition of HA, we are seeing that is the right one because it's in the correct size of the naked rat. We still see a little bit of the endogenous one, but it's present. This also modifies the architecture of the ovary because when we analyze collagen levels of fibers by PSR, we saw there is a drop in the collagen in the ovary, but also we can see by RNA-scope using a specific probes to detect NMR-Has2 in the animals that is highly expressed. However, this is an entire ovary, and we want to know exactly the effect because the other one can be confused by affecting other parts of the ovary. So for that, we breed these animals with DDX4, that is PASA, and will be in germ cells starting from PGCs, and NR5A1 that we're going to use SF1 as the easiest one, and it will be in somatic cells. We breed them, and then we analyze if they are expressing the transgene of the naked rat. And we can see here that this is Has2 in mouse, then this is like the negative control for NMR-Has2, nothing, and then it increased highly in the transgenic mice. We confirm the presence of these specific transgenes using NMR-Has2, a specific probe that we can see here in the DDX4 Cree driver and pink, and then the endogenous one in green. And here at P10, we're using the DDX, the SF1 Cree, saying that we are expressing it, we can detect it, but the next step is like, okay, you need to show that actually is getting to the ovary and loading and it's on the right side. So we have a postnatal day ovary 1 in mouse with the DDX4 Cree drive, SF1 Cree drive, and we can see that there's increase of the HA in the ovary, same here, but also that we are able to detect the different types of very high, well, the naked rat HA here in the right size in the transgenic mouse, and yes, conserving a little bit of the homo endogenous one. So the big question here is like, okay, this is really cool, but why do I care? And it's, well, we are trying to protect this kind of environment. And the real question is like, if we're able to mimicking what is happening in the naked rat. So for that, what we did is to get the ovaries of different ages. In this case is the Cree driver DDX4, and we stay with HA binding protein in green and in pink SOX2. We have the same experiment for OX4 and BLIM1. And what we can see is that in ovaries from mouse that express the naked rat transgene, we can see expression of these pluripotency factors. They drop with age, similar to what is happening with naked rat, but we keep going here to analyze how long they are present and how they are behaving. The same for the case of the transgenic animal with SF1, where we have this is surrounded by HA, and then we have these clusters that look almost like a nest or like a cyst, and present into P10. Again, is there present there and go drop down, but is present. Although I don't have enough time to explain this, we are analyzing how is this affecting the ovarian reserve. And so far we can say that actually seems like the ovarian reserve is larger, but also something really important seems like the later sites can be bigger in the animals that are transgenic for HA, but also it extends for how long they're reproductive active or they're having babies. Although those experiments still like work in progress, it seems like actually will be really useful and for develop new druggable targets that we can trigger to modify some of these factors. With that, I would like to say that why we should care about HA, and well, we should care because it's important in other parts of the body, but at the level of the ovary, at least in naked rat, seems that it's required to maintain the PGC population. And the levels are directly related or similar related to the enter into meiosis or the differentiation. Seems like even if it's mouse or human, the ovarian expression of HAS2 is conserved across the species. And this is really surprising for us because we were expecting that as is in adults, HAS3 will be the most important, but it's great to have this conservation. And also because the expression of the transgene of the naked rat in the mouse ovary seems that it's mimicking what we observe in the naked rat. How large or how impact thing will be the phenotype is a work in progress, but we are really excited about this. And with this, I would like to thank to the people that actually did this in the lab, Patrick Walsh, Ode, and Andrew, the different people that share with us reagents, technology, and animals, our funding sources. And with that, I will like to take questions if you have some. Thank you.
All right, thank you, Miguel. Very cool stuff. While the chat populates, I guess I'll ask you maybe a technical question. What are the challenges with maintaining a colony like this? And because you've worked with mice, you worked at different model organisms. What would you think is like the number one surprise?
I think that all of us we grow up like working with mice, so we think we have our brain prepared for mice, but we don't have it for new species. So these animals, a colony is just like one queen, a certain number of animals. So in the lab, for example, we have close to 60 colonies, and they live together for, you don't need to win them because they live like that. For at the beginning for animal training things, and I cook is like, okay, how it works. They live in a different temperature. They are like 28 Celsius. They have like high humidity. They don't need water, so they take all the water from the vegetables. So it's like a complete shift, but also give you the opportunity to try everything, and they're like so chill and fascinating.
Cool. Yeah, so I guess don't be afraid of trying new organisms, especially because it's probably a better model for human development anyway. Okay, so now let's get to the questions. We've got, does the oocytes quality is the oocyte, oh, sorry, Anonymous is asking, does the oocyte quality, is it the same between the young and the old naked mole rat? For example, the genetic mutations occur in the number of chromosomally abnormal oocytes.
Okay, so we have some a publication about the difference between queens and subordinates and how they're like they change in size, how they progress into in vitro maturation. So far, it seems like there's no difference in the quality of these ones. However, remember that I'm talking about that maximum amount of all this animal that we study so far is 10 years. So if we want to compare to what is happening in human, we need to wait a little bit for another 20 years to analyze. But so far, it seems like it's not. However, I need to say that when you push the entry into meiosis, dropping the HA, they show a lot of misbehavior during meiosis. So you can see problems with asynapses, problem with keeping the DDR machinery where should not be, and things like that.
All right, so we've got a whole bunch of questions. So Miguel, I've got a note that says we have 3 minutes. So try to give us 1-sentence answers for these ones.
Correct.
So the next question from Alka is, have you checked these genes in mole rat and mice only?
No, so we do always across the species, mouse, human, and naked rat.
Got it. Okay, next question from Anonymous. Great talk, thank you. You showed your data from a 6-year-old queen. Did you look at different ages of the queens? If not, do you think you would see differences in older queens?
Yes, so far we analyzed up to 12 years. Seems that there's no difference in this.
Great. All right, we've got 3 more. We're doing great. This is like okay. Does NMR use the same cues as musculus to enter meiosis? Does your single-cell RNA-seq suggest that unique aspects of NMR oogenesis relative to HA's role is intrinsic to the germ cell response or due to changes in cues from the somatic environment? This is from Tony DeFalco.
Tony, it seems like the first part, the meiosis, there are like a lot of shared genes that, or the most common ones, but they're like a signature that could be different. We are, and the second part of the question is something that we actually working right now.
Perfect. All right. We got another one now. Okay, we gotta be fast. Really fascinating presentation. This is from Pete. I can't see the last name. Pete. You described the germline as PGCs aside from expression data. Do they show other features of PGC, such as derivation of pluripotent stem cells, transplantation to produce teratomas, or migration?
Well, naked mouse don't produce teratomas, no matter what. Even if you do induce pluripotent stem cells, we culture this in vitro and we can expand them up to 5 times, and they differentiate into folliculogenesis. It's already published. And then we are working in how to differentiate that into IPS. Well,
All right, we're doing.
stem
We got another
cells.
question. 3 questions, 2 minutes. All right. Have scientists created knockouts for specific genes responsible for premature ovarian failure in the naked mole rat? And what are these results? Can this rat model delay the ovarian aging in a knockout?
There's no naked rat mutant animals. We are working on that, so hopefully you will have some NMR mutants soon.
And that was from Rima. Rima, great. Final question. Okay, we're going to do it. We got 1 minute left. HA is conserved in human. How do you explain that we lost this property of longevity? Do you think that they have a different role in the human?
I think that the role is similar. What is different is that there's this the gene stop expression in during fetal life. Meanwhile, in the naked rat, still like present. What are the regulators to say stop producing or expressing Has2 is something that we are working on.
Wow, we did it. All right, we got all the questions and we have a couple seconds to spare. All right, congratulations, Miguel. We all did it. All right, so if there are any other questions, please reach out to the speakers. I know there wasn't a lot of time, but I think this is fun. And with that, I don't have any other announcements to make, except that we have this Rising Star series. So check your emails. This is an ongoing series. I think we have 8 Rising Stars to get all together. So it's 1 o'clock. I just want to congratulate and thank our speakers again. And everybody else, take care and have a great rest of your day.
Thank you all.
And I'll stick around for some post-game synopsis.